This session is entitled Quality Management Systems, QMS. We are pleased to have Sam Haidar moderate this session. Sam, you've recently met earlier this afternoon. He is Senior Science Advisor at the Office of Study Integrity and Surveillance at FDA. Additionally, he's also a proud alumnus of the University of Maryland. Sam is also our first speaker, and Sam will be presenting on QMS study integrity considerations. Sam, thanks for moderating and presenting.
Thanks, Jeff, for the nice introduction, and greetings, everyone. This presentation will go over quality management systems for IVRT and IVPT studies, looking at study integrity considerations. The focus will be studies supporting bioequivalence determination, and what follows should be viewed within the framework of quality systems that we encourage sponsors to follow to ensure data quality and integrity. So this is our standard disclaimer. I'm sure you have seen it many times by now. My talk will cover overarching expectations for the IVRT, IVPT studies, as well as some specifics for each type. Many of the elements you've already seen in the prior presentation, so they will be repeated here. Elements which are important for the review and assessment team, and finally, reserve sample requirements and their importance. As would be expected for most regulated studies, good documentation is essential. Recording procedures, events, and observations as they occur, maintaining records which are adequate for the reconstruction of the study, the use of validated methods for sample analysis, and maintaining personnel training records and qualifications. Drug accountability is very important. Standard operating procedures and study protocols should be followed. Deviations need to be noted and justified. It's also good practice to have SOPs with objective criteria for repeat testing. Samples should be collected at time points specified in the protocol. Stability established during method validation should support sample handling during the study. The testing order should follow what's recommended in the relevant guidance. For multi-station diffusion cell systems, test and reference samples should be included in each run. Before the drug product is loaded into the donor chamber, the membrane for IVRT or skin for IVPT should be equilibrated with the receptor solution. This may not be feasible for the Hansen microarray system, and we recognize this. Instruments need to be qualified and calibrated, as has been mentioned many times previously. In cases of repeat testing, there should be scientific justification. Otherwise, the original results should be reported. So next, we cover items specific to IVPT studies. To help reduce variability and possible bias, test and reference products should be tested on skin from the same set of donors. A balanced design should be followed, although there may be exceptions, as was discussed earlier on day one. There is a scientific approach to dealing with unbalanced situations. As stated previously, instruments and methodologies need to be qualified and validated. For these type of studies, excised human skin appears to work best. It's important to include non-dosed control from each skin donor and to collect a pre-dose zero sample from each diffusion cell. Conditions for the conduct of the pivotal study should mirror those of the validation study. Next, covering IVRT studies. Experimental conditions, including artificial membrane, should be the same in the pivotal study as was used during validation. Adsorption or binding of the analyte to the IVRT membrane should be evaluated during method validation, although initial assessment can be done during method development. The membrane should be chemically compatible with the receptor solution. Dose depletion calculations should follow the recommendations in the relevant guidance. The following considerations are important for the review team when evaluating IVRT IVPT studies. Module 5.3.1 is the electronic submission. In the electronic submission is the appropriate location for submitting reports. Method development, method validation, and pivotal studies should be submitted in separate reports. Also, separate reports for the analytical method validation, SOPs, and study protocols. Reports should include experiment dates and information related to the principal investigator, study management, personnel roles and responsibilities, in addition to raw data and chromatograms. Also, the reports should include information related to quality control, quality assurance, personnel responsibilities, and the archival of records. Ideally, records are archived in a secure location with limited access and protection against fire, flood, and pests. So this is applied to paper documentation. Now, shifting gears from QMS considerations to regulatory requirements. Reserve samples are required for in vivo bioavailability, bioequivalence studies, and for in vitro studies supporting VE determination. The requirements and recommendations are referenced in 21 CFR 320.38, 320.63, the guidance for industry published in 2004, which, by the way, may be updated in the near future, and in a compliance policy issued last year in August. So looking at 21 CFR 320.63, which is specific for mentioned in vitro bioequivalence study requirements, it states that the testing site, whether it's the applicant or the CRO, contract research organization, is responsible for retaining reserve samples of the test product as well as the reference. Reserve samples should be retained for a period of 5 years following the approval of the application. And if the application is not approved, then for 5 years after the end of the study. These are explained further in 320.38. From the inspectional point of view, some of the most common deficiencies observed in vitro BE studies are related to reserve samples. In many cases, reserve samples were not retained or they were not properly handled. For example, they were retained based on CGMP regulations rather than bioequivalence. In many cases, this has resulted in the repeat of the study. An important point to keep in mind is that if the in vitro study is used to support bioequivalence determination, then bioequivalence regulations apply. If to meet other requirements, for example, CMC manufacturing, then other regulations may apply. Here are some general recommendations with respect to reserve samples. Reserve samples should be randomly selected and retained from each shipment. Quantity-wise, typically for multi-dose products, 3 units of the test and 3 units of the reference from each batch tested. Reserve samples should be stored in their original containers in a separate area with access limited to authorized personnel. Storage conditions should be consistent with the product labeling. So to summarize, this presentation covered important considerations for IVPT, IVRT studies with focus on those supporting BET determination. It included general recommendations from the inspectional as well as review team perspectives. I also tried to highlight the importance of understanding the requirements related to reserve sample retentions and their importance. Finally, I would like to acknowledge the following individuals from my office, OSIS, as well as from the Office of Generic Drugs, who made valuable contributions to this presentation. And I thank you.